This is my fourth block class, the one that meets directly after lunch. Today we are on the last part of our unit where we are talking about what's the difference between gel electrophoresis, plasmid-based transformation, and DNA fingerprinting. We do have a little activity that we wanted to do. We have a lot of things going on this week. And this is the last week in our 9 weeks for this quarter here. So our essential question for today, what's the difference between gel electrophoresis, DNA fingerprinting, and plasmid-based transformation? How do I conduct an experiment to demonstrate each of these techniques? So our indicator is HB4B2. Obtain, evaluate, and communicate on how biotechnology may be used in the fields of medicine, agriculture, and forensic science. Now, the only thing I want you to do again is to get down the 2 essential questions.
One of your classes, so always pick up my phone.
I do have some paper over there if you need to get some.
I gotta do one of the little trade for trade thing. I ain't got time.
Right here.
Bring it back.
Okay, now, the quarter is almost over. It's lined up in that area over this pack of paper.
Much is right there. Yeah, right there. No, you see the red button?
Yeah.
Go right.
Oh, yeah.
Please make sure you replenish your supplies. Thank you in advance. Now what I want to do, I want to go over 4 questions. I hope that you picked up your lab books. I see them still stacked over there by Ms. Dyches over there. Please pass the lab books back. Thank you. Now this activity probably will take the majority of the block. I will go over the manual with you. You must know your base pairing rules in order to do this activity.
Talking about A go with you, you go and stuff like that.
Okay, when does A go with you?
When you see A? No, A go with you when it's a U.
A go with you when it's RNA.
Thank you. I guess that could have been interpreted more than one way. So A goes with you when we're talking about RNA, but A goes with what when we're talking about DNA? T. Thank you. So what you're going to be doing, you're going to have to create the strand, and they will tell you which nitrogenous bases that you need to use. You're going to have to match those up, and it goes step by step to tell you that. Now, when you get to the part in the lab where they tell you to tape down, we're not going to tape down anything because we have to continue to reuse these kits, and you have another classes coming in here after you. So I will show you how to do whatever. You will work with a partner. The lab stations are already set up. But again, I'm not going to tell you what to match, when to match it. But I will be coming around and asking you different questions. G pairs with who?
C.
C. Thank you. Remember that. And they will tell you how to organize what needs to be organized. So what I would like to do, I'm going to go ahead and skip some things so that we can get started. Can you pass these out for me? In section of the first part of your organized lab books, you're going to go ahead and turn to the table of contents for me. And I'll share with you some background information. Please don't write on my sheets because I have to reuse the sheets. And please be careful with what you are doing. So now, If you look in front of you, you have the lab aids number 75, DNA fingerprinting, diagnostic, replication PCR, and RFLP. Today we're going to do part 1. So what I'd like for you to do is to put DNA fingerprinting, diagnostic in your table of contents. So turn to your table of contents. Turn to your table of contents. Look at the very top part of the lab. You have the title of today's lab. So what I'd like for you to do is to put the lab title. Here you go. Put your title of today's activity there for me.
Can you repeat?
Okay.
You said
Okay, wait a minute. Seems like I'm leaving some of y'all behind.
DNA on fingerprint diagnostic.
On the first page, at the very top, it says in bold, Lab Aids number 75, DNA fingerprinting diagnostics. I want you to put that in the table of contents. Now you know what page you began on or you ended on. Please record that in your table as well. Has everyone done that? Okay, now turn to the next clean page in your lab sheet. So what I'd like to do is talk about the introduction. Now, we always have 2 paragraphs for our introduction. What you need? Oh. The first paragraph is what? What do we normally put in our first paragraph?
Tell
Your background information. What are we talking about in today's little activity? So we're still on our biotechnology, and our biotechnology is telling us that we're going to be doing the PCR, and we're going to be working with DNA fingerprinting. Tony, can you read the first paragraph for me?
right here.
Yes.
DNA fingerprinting or profiling is a diagnostic tool with many applications. Identifying genetic diseases and disease-causing microbes, establishing paternity, plant and animal sciences, crimes involving wildlife poaching, and the identification or exclusion of suspects in criminal cases.
So they can take your DNA and they can do all types of different things with it. So today we're going to practice looking at a specific strand of DNA, and then we're going to see possibly what could happen if my DNA is not copied correctly. So today we're going to key in on a specific disease or disorder. So we're going to read a little further and see what we're talking about. Miss Tyler, can you read for us?
DNA fingerprints may be obtained by 2 different methods, PCR and RFLP. Polymerase chain reaction, or PCR, is a process whereby specific pieces of DNA are copied in a short period of time. This process takes a single copy of DNA and uses it to create millions or billions of copies of itself. The process takes advantage of the cellular components necessary for DNA replication, and also takes advantage of animal properties of chemical bonding in the DNA chain. The other technique involves cutting the DNA sequences rather than copying them. RFLP, restricted fragment
Polymerase.
uses special enzymes called restriction enzymes that recognize specific sites in the DNA molecule. The molecule is cut at that specific site only.
Okay, so we're going to stop there. Now, your background information. You can take those 2 paragraphs and pick out keywords and phrases and summarize to create your introduction for today's activity. In addition to that, I have up here some history and the steps in today's activity. So you're going to be constructing the model of the DNA. You're going to have to base pair things correctly. Again, if you don't know the base pairing, you're going to have to go back and review, but we did do that shortly. A goes with T, G goes with C. I will give you a couple of minutes at this time to go ahead on and write your introductions. Please write your introductions. Remember, the first paragraph will be your background information, and your second paragraph will contain information concerning why are we doing this activity.
All right, first I got this.
Go ahead and do your introduction, yes.
Oh, right, first and second paragraph.
Okay. Look at what's on your sheet. Now, what goes in the introduction? What does that mean?
The first paragraph.
What information, what types of things do you put in your first paragraph? How do you write an introduction? You forgot?
No, I mean like,
Okay, so what are you saying?
hold on, are you confusing me, right?
Where did I confuse you? I'm sorry, where did I confuse you?
I was asking like, take stuff out of here. Because
Yes.
Oh,
Mhm.
all right.
Because when you do your introduction, it's always, tell me about, in that one, what could you put in there to tell you, if I didn't know anything about DNA fingerprinting and PCR and what could I put in there to let a reader know that you're about to conduct this experiment and you're giving them this information? So that's how you're going to approach writing your introduction. What new terms could I put in there? Do you see anything in there that's unfamiliar to you? Okay, so put that in your introduction. You have a question, Tony? Oh, you look like you. Now, it's no right or wrong, but you need to inform me, what am I doing today? What does this have to relate to the indicator? What could I say about DNA fingerprinting? How was it used? And remember, you only have to do 3 to 4 sentences in your first paragraph. I'll give you 2 more minutes. You have a question, Mr. Chisholm? Can I help you with? That scared you? Why did it scare you? It's just the Elmo projecting what I have underneath here. This is just some extra history and some background information. I'm not going to go through all of it, but I'll share some of it with you. And you might remember some of these names and things from your timelines. I'll give you one more minute, then I'm going to ask you to share what you wrote, some of it. Okay, Miss Dyches, can you please share with me? You don't have to be shy. Share with me what you wrote. It's no right or wrong. If you can't get it wrong unless you know you just don't try. So share with me what you said, please. Can you please speak louder because I can't hear over the air?
DNA fingerprinting or profiling is a diagnostic tool with many applications. DNA fingerprints may be obtained by using two different methods, PCR and RFLP. PCR is a process whereby specific pieces of DNA are copied in a short period of time. RFLP or restriction fragment
Polymerase. ASE is an enzyme. Okay.
uses special enzymes called restriction enzymes that recognize specific sites on the DNA molecule. In this lab activity, we will be constructing the CFTR gene in its normal and nucleus.
Okay. Let me see. Somebody else. Keyshawn, I want to know what you came up with. Okay. Just share with me what you have so far.
DNA fingerprinting, to begin with, DNA fingerprinting or profiling is a diagnostic tool with many applications that identify genetic diseases and disease causing
Microbes.
Also, crime involving wildlife poaching and the identification of inclusion of suspect in criminal cases.
Okay, stop. Now, you're reading directly from the sheet.
So you got.
Did you add anything to that? What does that mean to you?
Oh yeah, I just want to finish.
Okay, but tell me something that you got from what they're saying. What are you going to be doing today? Okay, working with different DNA. So how do you think you may be working with different DNA? Thank you, the different strands. So back there, you have a bag with different nucleotides in it with the different bases, and you're going to be putting them together based on the order that they tell you. So today you're going to be working with the gene that's coding for cystic fibrosis. Now they tell you what a normal gene would be, and then they tell you what a mutated gene would be. Up in front of you, you have the different people that have made contributions to the study of DNA. So they start talking about Watson and Crick. Start talking about the history, who discovered what. 1953, they came up with their model. 1955, they start talking about the enzyme that worked with the DNA polymerase. 1972, they start talking about Paul Bearing and the research he did on cloning. So it's a number of different things that we could use to go back in history to help us contribute to what we have now. So I'm going to move on. What is the purpose behind today's lab? What do we put for that? Anybody? Tony? What are we doing today? What's the purpose? What's your second paragraph?
You get a closer look at the types of DNA.
Okay, so you're going to look at the different types of DNA. Now, based on what I said, were we paying attention? What are we specifically looking for?
DNA strand.
What gene were we talking about? Okay, and that will tell us what? Cystic fibrosis. Thank you. So let's look and see what is cystic fibrosis, because we talk about that or we hear about that a lot. PCR is very similar to DNA replication. In the process of replication, the entire DNA genome or organism is copied. However, with PCR, only a small region, the locus of the DNA is copied. PCR can be used to diagnose genetic diseases such as cystic fibrosis. CFTR, which stands for the cystic fibrosis transmembrane conductance regulator, is a gene that codes for a protein found in the membranes of human cells. This protein helps to regulate the flow of chloride ions in and out of the cell. If the CFTR gene becomes mutated, again, mutated, change, different, something is going wrong, thereby disrupting the function of the protein, the result is a disease we call cystic fibrosis. A thick mucus buildup in the lungs, reducing the flow of oxygen to the rest of the body and allowing for severe and deadly bacterial infections. Cystic fibrosis usually proves to be fatal by young adulthood. And I told this story before in another class. When we were growing up, we had this childhood friend, and probably about 5th grade, he could walk, he was healthy, but he had cystic fibrosis. So he lost the inability to function, and he became disabled to the point where he had to, function from a wheelchair. And then by the age of 18, where you could look and see him throughout, he just started losing control of his limbs, and he just couldn't really do anything for himself, and eventually got to the point where he was bedridden. And by the time he got to year 18, he ended up passing away. So we have a lot of things, diseases and disorders that happen to us and in the African American community that causes us harm. And again, it's because of a mutation. So today we're going to look at that mutation. What I want you to do next is go to the next part of the lab. What is the next thing that we usually put down? Okay, we did materials. When we go back in the back, you can look at what you have and write down what's on the kit, or you can say kit and mat, and we'll go through that. So hypothesis. Now, how do you think we're going to create this hypothesis? What were we trying to find? What are we trying to find? Look at your sheet. Cystic fibrosis. Thank you. Materials hypothesis. So, what do you think would be a good hypothesis? How are you going to create your hypothesis? What are you going to say? Remember, it's supposed to be an if-then statement. Yes, that's correct. So let me see what you come up with.
You gotta wait for your materials by then.
When you go back to the station, you can put your materials. You finished with your hypothesis? Oh, yes. It is. They gave you the definition of it. And then if you look down further, it tells you what the DNA sequence of a normal person would look like. So if they have anything other than that, then what would that mean? What are we looking for? Okay, so if they gave you the normal order or DNA sequence and you get something that doesn't have that, what does that mean? You don't know? If it's normal, I don't have it. But if it's abnormal, then what? You got it. So you have it. If I look at that sequence again and I match up something other than that, or if they give me something that's deleted, I know that I have a mutation. Something has not copied correctly. So what'd you come up with?
I'm trying to think of a question.
Hmm? Well, go ahead.
I was about to say if it's not human like, then it's abnormal.
Okay, so we are dealing with human, but we're looking at a human to see if the human has cystic fibrosis or if the human does not have cystic fibrosis. So how could you formulate that into a hypothesis?
If it has fibrosis, it's not.
Okay, so how would you know that if it was
After you test it, take DNA.
exactly? So if I look up here and they tell me to create this strand, and then I do my matching base pair, and then I come over here and I don't have those same letters in here, that means what? They have cystic fibrosis. Okay, so create your hypothesis. Anyone finish and like to share? So now you know I'm going to call on you, right? Because you've been in that position long enough. So I know you have mastered what it is that you're going to put on that paper, right? Tell me something. Help her out, please.
I said if the CFTR gene is in a normal form.
Wait a minute, you need to pay attention. Unless you have mastered it and you know what you're doing, but you need to pay attention. Okay.
I said if the CFTR gene is in a normal form, then the
Cystic.
cystic fibrosis disease and the gene is not in mutation.
Okay. What do you have?
I wrote if the CFTR gene is mutated, then you have cystic fibrosis.
Thank you. What do you have? Read it, please.
I think if the CFTR I think if the CFTR gene is mutated, then I think it is cystic fibrosis.
Okay. I need to hear from Miss Alexander. What do you have?
If the CFTR gene becomes mutated, then it would be the result of the disease we call the fibrosis.
Oh, very good, very good. Okay, so this is what we're going to do. Now, I'll go ahead and tell you, you can't do the lab unless you read through the steps because it tells you specifically when to line up the nitrogenous bases. But you yourself will have to match up the base pairing sequences to come up with the correct DNA strand. You will work with a partner. Everything is laid out. We're going to try, no, not going to say try, we are going to finish the activity and go over certain things. So, everything is set up. There's one station that I don't want you to go to, but 4 stations over here, 3 over here, and 2 over there. You will need your lab book. You will need this piece of paper. You may report to the lab, and I will be going over things with you and asking you certain questions as you build your sequence. Now, hopefully everybody put the things back in the bags like they were supposed to. But again, you cannot do the activity unless you read it. So don't try to go to step 3 when you know you haven't done step 1 because something is going to be wrong. And I will go over it in general with you. By now, Mr. Chisholm, everybody should be at the station. Now, what we didn't do, again, I told you when you came back here to go ahead on, look at your materials. So go to your workbench, look at what is in front of you, and you can just say that part of the material, the materials would be the kit or your contents of the bag, your nitrogenous bases. Are we ready? So we got a bag here, a bag there, bag here. What are you looking for? Oh, I'm about to say now it's not a necessity that you have a stool, but that's fine. Okay, now you need to get a system. There's no such thing of you're working together and one person is looking and the other person is doing nothing, right? So we don't want to see that. That's fine. That's fine.
Can I call this material post?
Okay, yes. So you have on your station your strips, your adhesive strip. Now always look at your sheet to know what you're doing, right? Okay, so I gave you a bag of kit, a kit with different bases in it. You have labels in there. So what you can do, you can go ahead and take the things out of the bag. Please note, everything that you take out of that bag, thank you so very much.
Ow, why you hit my knee? This stupid, bro.
Be careful. All right, let's move on. Now, again, everyone does not work at the same pace, but you must be working. So what I'd like to do is to go over procedure. Is everyone on the procedure part? So it tells us, I'll share with you. It tells us, it is possible to determine whether or not a mutation is present in the CFTR gene. You will be comparing a normal CFTR gene with a mutated CFTR gene using the PCR process. Number one, construct a small portion of the normal copy of the CFTR gene. The gene actually contains more than 4,600 nucleotides. Do you think we're going to have 4,600 nucleotides in that bag? No. So, your job is to just construct a small portion of what you're going to be looking at. In the following manner, using the longest adhesive strip. So, where are your adhesive strips? Find your adhesive strips. Okay, now, does everybody have their adhesive strips? What are you doing with your adhesive strip? Post it on the glossy side of the strip and line up under the title DNA fingerprinting. So if you look at your boards, at the very top, what does it say? Okay, thank you. So what you're doing, you're working underneath there. DNA fingerprinting diagnostics.
How many adhesive strips should you have?
What did your procedure tell you? We're going to do one thing at a time. What did your procedure tell you? There you go. So how many do you need at this point? One. See, you answer your own question. Okay, let's move forward. Line it up under the title of DNA fingerprinting diagnosis. The print side will be the top layer. Now, do not take the adhesive off of there.
Oh, okay.
I'm going to say that again. Please do not remove the sticky stuff because we're not sticking anything down because we have to reuse this for other classes. But you're still going the same process.
Now look at Shalique.
What did I just say?
Don't remove it, Shalique.
I did it.
Okay. So go ahead, follow along with the procedure. After placing the side of the board strip, this will reveal the adhesive part. Again, we're not removing the tape. So it says leave about 5/8 of space so that the label template normal CFTR can be placed. So what I want you to do is to look in your bag, find where it says template normal CFTR. Please find that. Everybody should be looking.
Is it going to be on the blue piece of paper?
Yes. Because what? You've got to label it, right?
What should it say?
Go back to the paragraph. Leave about 5/8 of space so that the label template normal CFTR. Did you find it? Were you able to find it?
Oh, CFTR.
Okay, we found it. So we found it. So put that out.
Wait, this one that says
What does it say? That's what you're looking for.
They got COPIA.
Yeah, I told you that.
Okay, we want the normal.
So this is it right here.
Yes. Okay, now, you don't have to wait on me. Go on to the next part.
Oh, it's two of them.
Look at that long strip. Does everyone see the DNA sequence?
This right here.
Yes. Did we find the DNA sequence?
Hold on, I need to find out where y'all at. Y'all on the number 2?
We still on number 1.
Okay.
Leave about 5/8 of space. Did you see that? You don't
I
see
see
that?
now.
Okay, now look underneath there.
Okay, by the A.
Yes, that is what you need to build. So at this time, please build that. Practice building that.
Was it right on it?
So,
No,
why
we're
would you
supposed to
write? You see all
build.
these letters?
We
Why
got
would you
all
write?
these letters.
Oh, so you put
So you got to build the model.
put this right here like that.
Yes.
Up under that.
Put it on top of it. Yes. Oops, sorry.
Use all of the
Okay, now the question you asked me, use all the letters. If I just said build the strand, what does that mean?
Use all of them.
You got to build exactly what you see. You can't do anything extra.
Okay, so first put what's right here, and then
Then we're going to go from there. So remember, with your procedure, always do what it says. Don't deviate because you're going to cause a mutation before you are supposed to. So between you and your partner, make sure you have the correct sequence. Okay, where are you? All right. Yes, keep going. Very good.
top of the script.
It's true.
Yes, put it on top of the strip. Now remember, normally we would have peeled it off and it would have stuck, but we can't do that because we need to reuse the kit.
You don't have to. I mean, you can be on the class.
Well, I have 3 classes of biology, so
You only got one class after this, though.
I know, but the first class had to use it. If they would have stuck it down, then you wouldn't have been able to use it.
They would have been there and did that way for it. They would've just-
Again, make sure you have the correct sequence. It's easy to put the wrong base up there, isn't it?
Let's cut a piece of that paper, like cut a strip of that paper.
For what?
And then glue it on it.
Because if you glue it down, wouldn't I still need those bases?
It ain't
You
coming
got a
back.
lot of these.
No, you got to go through the whole process. There are 10 different steps that you need to do.
Like you could let us glue it on, then cut it off and glue it on again.
It's not an option. Thank you. Okay, what's happening back here? So what system do you have back here? System? What system? How are you helping? What are you doing?
Oh, ma.
Mhm.
She's reading me out the thing.
Oh, so you're reading and she's putting them down. Okay. How are you helping with the process?
I'm flipping all the letters around. Got him all right.
Oh.
What color you need? What letter you need? Tell me what letter you need. I'm gonna have it for you. T? Yeah, you know the letter. G, G.
Start sliding down. I know who we at. We at C. No, I thought I was looking to see if we supposed to put all of them. Oh no, it says a small portion.
So why do you have that space there? Why is that
We
gap
were
there?
sliding it over.
Oh,
Because
okay.
we thought we had to put all of them. But it says a small portion.
Okay, but you want to build the entire strand, right?
So what this say small portion for?
Okay, but go back to what it said. How long did they tell you the sequence of that gene is normally? Remember I said we don't have 4,600 pieces in this bag, but the portion we have, we're just going to do this. So when they say small portion,
thought they
this
meant
is
this was
no,
small.
no, that's not what it's saying. Okay.
It has to be straight.
You're gonna be moving stuff around. So how many more minutes do we need? 1, 2, what?
After we do it, we translate.
Okay, now, very good. So, but look at, before you do whatever you do, look at what it's saying. As your workboard indicates, the strand you have just made is a specific 5' to 3'. Now, what they mean by that, if I hold my board up and I look at the top, what numbers do I have up here?
5 and 3.
Okay, so that's my 5' and that's my 3'. So now you're going to have to build the other side of the ladder, but now you're going to 3 to 5. In order for you to do that, Mr. Chisholm, you're going to have to do your base pairing rules, but you're going to match them upside down. Now, what does she mean by that?
The A
Like it is right
will
here.
it'll be like this, the 2 will be like that.
Yes. Okay, so what I want you to do now, and you shouldn't have to look off the paper to do that because you are the expert and you already know your base pairing rules right. Isn't that right, Mr. Chisholm? A goes with what?
T,
And G goes with?
C.
Build your strand. So you may get another long strip.
Answer for me because I know it anyway.
Oh yeah, you know it. So get yourself another strip and build your base pairing. I'm coming around to check. Yes.
Hold on, you said turn it out, turn it out.
Your first strip, that's fine.
What you mean, give another strip?
That means if your first two not
You use a first adhesive strip for the first one, right? So now you're getting ready to do the other side of the ladder. So you're gonna do the same thing. Get another strip.
And put it
Now
between it?
match up. You're on the second side of it. It should look like a ladder, right? So where's your next long piece? So you put that on. See, that was supposed to, well, see, that was supposed to go on top of there. And then now they're telling you to build the next base pairing. So what goes here?
Oh, the T. Oh, so
Okay.
these supposed to go on top of that.
Exactly. Yes.
So, Miss Fran,
Uh-huh.
turn it upside down and
You got to turn the letters upside down. Yes.
so
take it longer.
Leave
Leave
that
it
the
at
way
that.
it is, and
That's just the
then
way it
build your
is.
strand.
You went upside down like that right there.
There you go.
for the normal thing, put it right here.
You put that on top because your letters go on the adhesive strip, but you're just using the label to tell what it is. So if I didn't know anything and I was coming in here to look at what you were doing, I should be able to tell based on how you've labeled your model.
Which label we gotta use next?
So now I
What are you
translate.
doing?
I just said all of them.
You're building the other side of the model, right? So what do you want to put on there?
Which way do we do it? This way? Just do it backwards,
Yeah.
but just the same letter.
What do we mean by base pairing?
You do it. What goes with it? Like you go with A.
Are we doing U's?
No, we do a T.
So why you tell me U's?
Because I was just.
Okay, now your job is to help your partner, right?
Right, yes, ma'am.
So obviously your partner has some questions. So
Use this one.
Yeah, she
what are
didn't
you
ask
going to
you,
do to
though.
help her?
I'ma tell her
you
what to
sitting
do.
there.
A goes there.
It's a teamwork,
Put
right?
A, okay, and then we gotta put T. Remember T.
Okay, now.
Gotta put
Where
A.
we at? I done lost where we was at.
So that's just a label. So you can put that to the side. Now, what do you need to do? How are you? Okay, go ahead. Tell me what you're gonna do.
Like how, like A go with T,
Where we at?
G go with C.
So you need to find those out of your bag and put them in there.
Oh, here go right here. Ain't it?
And I said because you the expert, you shouldn't even have to look at that. You ought to go ahead on and match it, right?
Yes, ma'am.
Now you will use that to check it, but you're not supposed to copy that. Check your work
So when you put the label on?
when you finish.
Alright.
I haven't seen you.
Remember, DNA is looks like a twisted ladder. So something is wrong if you don't have something that looks like a ladder.
From the back, put camp.
T go with
I need a city girl.
Miss Brad, so what do we do next?
Okay, go on to the next step.
short one. Some up on your board. It's on the
Okay, check because yeah, there you go. Because everybody was supposed to have the same types of materials.
Next, label the
And
primers
so you-
on the left. Actual primers are made in the laboratory by the DNA
Synthesizer.
and can be different than in different sequences depending on the region of DNA to be copied. So wait a minute. Hold up now.
Okay, so notice where you are in your procedure. Now you're working on the next part of the PCR. So what is it telling you to do?
Make primers.
You got to make primers. So what are the primers that you're making? Look at what it says. You've done it. I want you to explain to me what you've done. So what did you do?
Made CATA,
Mm-hmm.
and then for the other one we made AATA.
Okay, very good. So now what are you going to do? Because primers do what? Read what it say.
Make the DNA molecules will target the region on the template.
Okay, stop. So it says the DNA will target the region on the
So
template.
this is
So what are you going to say?
so this is what's gonna tell us if it's
Cystic fibrosis. So now I'm picking out different areas on my DNA to target to start making copies. So your job is now you've got to match your primer to your DNA sequence. Where on the sequence would you put the matching CATA and AATA? So look at your strand. Where would they match up? Where do,
AATA right here. So do we put it in the inside of it?
how do you pair? Where do they go? It's going to be on the outside, right? Okay, so line it up. Where do they go? You got the top and you have the bottom that you must consider.
No.
Correct base pairing rules.
No, that's CT. You gotta be that way to just be crooked. No, I don't know. Over there. Over yonder.
Make sure you read it. Are you turning it the correct way?
But no, like it's gotta be different.
So where is the AATA going? Where is the matching sequence?
Did we do it wrong? CATA.
Go back to what it says about base pairing rules and figure out where it goes.
Puppy boot.
Miss Rainey,
Okay, what's happening over here?
what's this one right here?
Huh?
Complementary strand?
Okay, so you made the complementary strand, right? Because you had one side of the ladder.
So this one gonna be complementary?
Okay, now what part of the procedure are you on?
Two.
Two? Okay, what does two tell you to do?
Two. That's complementary strand. We just did it.
Okay, so you build your complementary strand. What's next?
And then we're on three now. So like, you gonna do some. You would need two more short strip pieces.
The shortest
They
piece?
say
or
two
like-
more pieces of single-stranded DNA primer. These DNA molecules target
Wait,
the
like-
region of the temple.
Short strip has leaves?
Mm-hmm. That's correct.
Yeah, you gotta make CAT.
Save it. C A
TH.
T.
Yeah, that's for the
T
first
A
one.
as in T. That's why you need this.
Okay, so Keyshawn, what are you doing? Which one are you?
I'm still on the.
Wait.
You just put the letter, matching the letter.
There.
Is that correct?
Hold this.
Okay. So how many more do you have to do? Oh, you almost finished. If you have already built your complementary strand, you need to move on to the next step. Remember, we have 10 things that we have to do. Don't just sit there. You need to keep moving. Move on to the next step. You got a partner. You just can't sit there and look.
Okay, no, that's the
Except for
shortest we
me.
got right there.
Are you sure? Did you look amongst those
I
things
didn't know
there?
any of us gonna be in the bag.
It's all those things are in the bag. Yes, I put the shortest things in.
These are too short.
Go back to the definition of primer. What did it tell you a primer was?
These things right here.
Are you reading on the paper, Sine?
I'm going with that.
Thank you. I said you can't. I told you what you're gonna do, but you can't do it without reading. Now, what did it tell you about a primer? Where are you in your procedure?
Step 3.
So step 3 says what?
Two more short pieces of single threaded DNA. So these,
Exactly. Thank you.
but not the long ones, but yeah.
Okay, so now you found what you needed. You answered your own question. Now, what do you do next?
Group them by 4.
So you've got to build the primer. How are you going to build the primer? How are you going to build the primer? So you have, yes, what did it tell you to do?
Some unused nucleotide. Oh, so get some
Okay, so you've got, here are your nucleotides, right? And now what did they tell you to do? You've got to make primer 1, wait a minute, and you've got to make primer 2. So here is what you will use to make your primers. So go ahead and make your primers.
and put CAT.
You got to do exactly what it says, because if not, you're going to have a mutation. Are we confused about base pairing rules? What's
We only
wrong?
got one little short strip.
Okay, so this
Shaq was trying to spell something out to me. I didn't know what he was trying to spell.
But what do next, Miss Branch?
Okay, what number are you on?
3.
3 out.
Okay, so 3
Check
we're
what you
talking
spell.
about primers, right?
It's what?
Okay, this one.
What
So
is-
you got your primer, you have your primer. You still need to build your primers. And you're building your primers on the short adhesive
So
strips.
that means I'm still on number 2.
We only
Are
have
you
one
sure?
short.
Y'all have 2 because I see the other one.
No, where at?
Look in that
Over there.
stack.
No more Miss Branch. Somebody jack her.
See they?
Okay, thank you. See, you've got to look for your
Yo, you
materials.
smart and missed the whole thing.
listen,
It was about
All right, we're on the same thing, so I'll just do this here at the same time. So now you're to the point where you're trying to make your primers. So they gave you a strand to tell you what primers you need to build, what primers you need to build. So look at the sequence for your primers. Find your nitrogenous bases. Find your nitrogenous bases.
What's that?
I'm not gonna tell you that. You find your nitrogenous bases.
Hey, Kanye West hard in it.
Which way the
What letters did they tell you to use?
Hey, you listen to Kanye?
Find them.
I'm
Well, you're going. It doesn't matter where you put them because you're going to have to match them to the areas on the DNA sequence. So build your primers and then match them to where they will fall on your sequence. Same thing for you.
Okay, when I do that, okay, you don't have to have a little one of these.
Don't worry about that. Build the primers.
Oh, so all you gotta do is put these and these letters right there.
Exactly. Okay, y'all need to move on. What are you doing? What's the question?
Okay, so these are the one that stands for that.
That's all hard.
Primer 1, and I got primer 2.
Are you paying attention?
So
Okay.
we moving on to Islam report, right? Because this right here just give me information.
Okay.
So we move on to the report. It says dissociate the DNA strands on the stripes as though they were being heated to 94 calcium.
Keep going.
Catalyst, whatever this
No.
word is. I said word right?
No.
What is it?
What do you think that is? It's Fahrenheit and what else?
Calculus.
Celsius.
Celsius. How you get calculus out of Celsius? Okay, go ahead.
Okay, it said, to help you do that, dissociate the DNA strands on the tribes as though they were being heated to 94. So,
Okay, stop. So
94
what they're
degrees
doing,
Celsius.
they're trying to get you to picture what is happening in the process. You're not going to actually sit here and physically heat anything, but they're telling you
Why?
after you heat it, it's
You got
going
burners
to
in it?
pay attention. After you heat it, it's going to denature, and so when it starts to denature, it starts to break apart. Yes. Yes. Okay.
No.
So
Well, you about to
after,
be in the bind.
and see now you on your own. So after it breaks apart, your job is to put it where it needs to go. So because you already know the base pairing rules, you need to take the primers and match them to the strand. So where would you put those two primers? That's what they're asking you. Where would you put those two primers? You got to take them and move them. I can't tell you that either. By now, you told me
I
you
got to
know
move. I got to move these.
you have to move the whole unit.
Now you know when I move these, this gonna fall off.
Just the letters. That's what. Just take the strips and move it where it goes on it. So C is gonna go with what?
Miss Brett, for primary one, do we
A
put
is gonna
Salvador
go
2
with-
in one?
T.
T and
Miss Brett,
what's that?
when they say primary one, do we just put the C word in number 1 and 2?
Primer 1 was the first strand that you built, the small short one. Primer 2 is
But it got C, it got Salvador.
yeah, that's fine. Yeah. Okay, so where are you gonna put that?
Wait, like what order?
You have to match it up to the correct base pairs. Where would the correct sequence be for that?
Miss Branch, did we be able to
Right here?
finish this whole thing by time class over?
Probably get halfway
I'm
through.
not understanding exactly what you asked me.
Okay,
I
see
understand
how
how
I
to do
have
this,
this,
but I'm just not-
see how you have CATA?
Mm-hmm.
What are the matching base pairs for CATA? What letters would that
So
be?
I got to
How,
move this down here.
listen to what I'm saying.
Oh, C
What would the
it,
letters
it'll
be?
be a G
Uh-huh.
and a T,
Uh-huh.
a and a T.
Okay, now based on those letters, tell me, do you see those letters in that order in any one of those strands?
Anyone at the top?
Yes. Where do you see those letters?
This would be a G.
It has to be in order.
This would be a T. This would be a G. And that'll be a T.
Mm-hmm.
This would be a G. And that'll be a T. And that'll be a.
T goes with?
No wait. All right. That's that. That's supposed to be a T.
Uh-huh.
This a A.
Uh-huh.
And this a T.
Exactly. Now find that
But
on that ladder, on one side of the ladder.
This is a G though.
So that doesn't go there. Where does it go?
All right. So that's C. I mean, all right. C. A. No, Miss Branch.
You can turn it upside down. You just need to find where it goes.
So it do
Miss
this.
Branch, I don't see it. Oh wait, can it go like start from here and go this?
If you can't find it, then you've done something wrong in your original sequence. Are your letters correct?
Uh-huh.
Well, then you better find out where it goes.
So do you do the same thing here that you did?
Okay, so exactly. So find out where the
Oh
match
my
is.
God.
What's wrong?
It's not on here.
You sure it's not on here? Did you put what it said?
C, wait. C, no, C, A, C, A,
You sure it's not on there? You can't match that anywhere on those strands. You better look carefully.
You better put, ain't you supposed to put G on the C?
Yeah, G,
Okay, you go ahead. Find yours.
A, T. See, that's supposed to be a T. So that means that's wrong, right?
Maybe it doesn't fit in that spot.
All right. So what I'm saying is, I'm trying to read it this way. Can I start from this G and try to read it back this way?
You can. It wouldn't go there because what would have to be there?
A C.
A C. That's not a C. That's a A.
Mm-hmm.
So where is it going?
He was asking Tim, he said Brian Cotton.
That's not this. That's right here.
So put it.
You didn't put it. Put it where?
If you said it goes there, then take your strand and put it where it goes. Where did you say it
It's
go?
over here on the top.
Okay, then put it over there. You got to move those and put it where it goes.
She gonna find out everybody knows. They telling everybody. Don't do it. Tell it.
Are you doing what you need to be doing over there?
Yeah. Are you doing what you
Did
need to do?
you okay, it didn't tell you to rebuild anything, did it? It said take it, what part of the procedure are you on? I didn't I say you can't do without reading it? You've got to take that primer and match where it says A goes with what?
You? I ain't see T.
Are you sure? A goes with what? Thank you. What you said A, you said I mean, you got to be sure about what you're talking about. So A goes with T, so you need to find out where it's going to be T, T, A, T. Where do you see that on your strand that you've already built? Where do you see that? You got to find. Now I'm not going to find it for you.
Come on here.
Yes. Because,
Right here.
so that's where you need to move that. Thank you. Okay, now whatever you have on that letter, you need to do the same thing with your second sequence.
So this still gonna be this thing too?
Mm-hmm.
Okay.
You gonna get over there and finish what you're doing.
I can leave at work, cuz.
Okay, back here, what y'all
I
doing?
need to bring that down.
Bring what down?
Turned upside
Okay,
down.
ready?
You can. You've got to match both of those primers that you created. Where does it match on your DNA sequence? You've got to
I'm
match.
gonna turn it upside down and do it.
That's fine. Didn't you turn that upside down? That is fine. Nothing wrong with that.
Dissociate. You on focus?
Okay, are we ready to move on? Where are you ladies? What are we
I
doing?
had a question.
What was your
The
question?
pattern. Did we do this right?
Okay, so tell them what you did.
We turned it upside down because it matched that sequence.
Exactly. Did everyone find where it matched?
Yeah.
Okay, so now y'all can go on to the next part.
But now with the dissociate, the DNA.
No, we
strand.
still, now we will copy. We still gotta do that, I think. Or is this that?
That was what you just did, yeah. Okay, now remember, you have questions inside of the procedure that you're going to answer, but you may want to jot down little notes so you won't forget what you've done. And just answer in a complete sentence, and that goes to part of the data and the observations part of the lab. But you are ready to move on to the next part.
So
This
step
will
five.
dissociate the
Yes.
DNA strand on the strips as though they were being heated to 94 degrees.
So spread them out.
Okay, so what they're basically saying to you is, next part of this procedure is where you heat it, and when you heat it, it starts denaturing, so you want to break it down because you want the DNA strand to zip and unwind. So that is what you're about to do now. But you're not going to physically do that, but what they're going to ask you to do is to kind of duplicate what would happen if you were heating the protein for the denaturization. So, okay, now just because I've left from over there, Mr. Chisholm, that don't mean stop working. You need to go ahead on to the next part. So, let's just do this together. Is everyone on number 5? Have you already matched your... You can put that in trash for me, thank you. Have you already matched what you needed to match with your primers? Okay, so that's fine. That's fine because remember you're supposed to change the direction anyway. Now let's move on for the sake of trying to understand what we're doing. So 4, it dissociated the DNA strands on the strips as though they were being heated. Again, you're not going to actually physically heat it, but they're doing that, or they put that in there to let you know that that's part of the process. And after that, because they've done that, something happens as a result of that. So we're about to see what the result would be. So carefully examine the primers. To what sequence will primer 1 bind to? So that is where you looked in your notes, or you looked at your sheet and you wrote down where you connected it. So please put that somewhere in your journal, what letters the primer is connected to. Again, please write what letter combination your primer is connected to
Write it in our.
in your lab book.
Do the right
Mr. Chisholm, you sidetracked again.
You can write that at the
Come on
end.
back, you're missing that part,
Can we
huh?
write that at, can we write that at the end of we do the whole procedure?
It goes underneath the data and the observations.
Yeah, that's what I'm saying, after
What
we
is
do.
this?
Extra stuff, you don't need that.
I'm gonna use them.
For what? You use what you have.
So just write down the letters that
Huh? Yes.
What is this?
You said you didn't have them, that's why we couldn't
Hey,
take
right,
the stick.
but that's what
Why
I'm saying.
put out extra stuff just to have extra stuff out? So you making
So it's GTA.
an observation about what was created and what is generated based
Write
on
down
what you
the primers and
the
what they
primers
pair
gave
with.
you. Thank you. Put that in your journal. Put that in your data and your observation parts.
What I supposed to do? I ain't even did the data allocation part.
What did I just say? You just made an observation, right?
She just said.
So put down what you said.
Observe your observation and your
Okay. You don't know what you said.
That's it.
So moving on, again, you wrote down your letters that corresponded with the primers. So now we're moving on. Note that before primers can bind to the template strand, the temperature must be lowered to 94 degrees Celsius. So you've already done that because you've attached where the primers are on the DNA strand. Moving on to the next one. Number 6. Everybody should be on number 6. The enzyme DNA polymerase is responsible for copying the DNA. It starts right where the primers end. The enzyme builds a new complementary DNA strand in the 5' to 3' direction. Therefore, it reads the template strand in the 3' to 5' direction. The enzyme looks at the next base pair where the primer ends and picks up a free nucleotide in the cell that is complementary to the base pair. It adds it to the primer, thereby extending it and building a new complementary chain. The DNA polymerase then looks at the next nucleotide on the template DNA and carries a complementary base pair to add to the growing chain. It repeats this process until it runs to the end of the template DNA. So that's what we're looking at now. So look at your little model. Use the DNA polymerase piece to make a copy of both DNA strands. So now they're asking you to look at what you've done, make a copy of what you've done. Now you have enough letters left in those bags and on your station to generate another model of DNA. Please, at this time, go ahead and create another model of DNA
The 2 strands right here.
based on what you have up here.
Need another long one.
The original strand, look at what's on the model. I want you to build that. Practice building that. What number are you on?
You need another one. Someone's at the door.
Please get that for me. Okay, so now you don't understand me, right? Okay, so look at what we just read. Do you have the instructions with you? Were you following along with me?
I think so.
You think so? Go over to the next page. Look at the top. Read that for me.
From the all the way at the top.
Mm-hmm.
DNA
Polymerase.
polymerase needs to make a copy of both DNA strands. Remember to build the strands in the
You just
5 to
answered the
3
question.
direction.
Okay, so what they're asking you to do is to build that model. So I said go ahead and build the model. You have enough letters here to build another model. Yes. Again, Some of you are getting lost, but the whole idea of it is to what? Make copies. So if I make a copy of something, what am I doing?
So
What is it going to look like if I copy something?
Exactly alike.
It's going to look exactly alike. So what are they asking you to
the
do?
data and the observation will go right here.
Huh? Yes.
It's gonna be a
Copy it. Thank you. Go ahead on and copy it.
Go ahead on and copy.
But I'm lost.
No, you're not.
Okay, explain to your partner. Yes. Practice. Yes. Hurry up. Stop whining. You just wasted 1-2 good minutes whining.
Second, she didn't talk now. 2 minutes.
You're wasting time. Yes. Thursday.
Thursday. What,
I'll talk about that during tomorrow. Thank you. Moving on. Moving on.
Ludacris. Yeah, all
So
that.
we supposed to be copying again?
No, the stuff.
What number we on?
Okay, how you explain that to your partner?
Well, so we on the same thing I left off on?
That's not a prime example, actually.
We're making copies of the original DNA strand. Go ahead on and make the copies. How would you make the copies, Alexandria?
Oh, which one? The DNA or the RNA?
RNA. Where's RNA? Why did you say RNA?
DNA, I forgot. I don't got no U.
Thank you.
See that? See that? And I don't do
DNA.
nothing. I don't do nothing.
Okay, so go ahead on.
Okay, so why this
You
right
making me
here?
nervous with that camera.
So why this right here?
Ms. Brent,
I'm gonna see you after the camera leaves.
At the camera leave. Okay, what's your question?
so I put for data and observation.
I don't think that's supposed to
Okay,
be.
so your data and observation, see those questions that you have in there throughout the lab? I said that your questions go along with data and observation. I'm not going to ask you to go ahead on and draw this activity that you did, but with each step, you had some questions that you should have answered.
So just answer them for the data and observation?
Yes.
Oh, I got you. So, all right, now I got to move next to move.
Oh, you was so little.
make my-
Okay, I don't see any movement here.
So now we got.
Now remember I said team.
On the
Which one of the
paper,
questions?
dog.
See those questions? Turn it over for me,
Yeah,
please.
wait a minute.
We about to come back and do the same thing.
You talking about 1, 2, 3, 4.
Exactly. Thank
So
you.
this one.
That's it.
Ms. Branch, I think I need a partner.
Okay, so why didn't you work with Miss Shalika?
I think you work with
You chose to come over here and sit by.
I thought
Why
she
didn't you
worked
work
with
with Miss Jayla?
didn't you
You
work
you
with
chose to
Shalika?
come over here and
Listen.
sit by yourself.
Because
So
I thought her and Shalik was working together.
I did too.
that was your choice.
Why do you work with Sadiq Wali?
I would with it if I knew.
Oh, say,
Okay, again, everybody should be making their copy of the original strand. Please do that.
Do you gotta put a label on it?
No, just do copies so we can move on, please.
No, this thing not long enough.
What's not long enough?
This little strip. We need some new ones out the box.
You just want something?
That's
Go
not-
ahead and use those.
It's not long enough, Miss Brent. It's gonna be all right. Look, it still ain't long enough.
You let it hang on the thing?
No, I ain't making nothing out. Miss Brent's not here. That's it. Everybody else gonna use the blade.
And you know what? Yours better be right. I'm
All
gonna
that yapping
call
you
on
doing.
you,
Right.
and you better be able to explain it to
We
me.
need 2 animals.
If you don't get it right, I say write them up.
then you are down.
How about you just teach on Mac Miller,
Huh?
Mac L. Miller?
You have a question? You don't understand? What is it?
No, we gotta do the
That's
bottom
right
one
there.
too, right?
Yes. Remember, it's double. It's a double helix. It's 2 sides for your ladder.
DNA is like way too much. I can't deal with it.
Yeah.
It came up my
So we put all of this.
nose. That's nasty. That's so nasty.
Mr. Chisholm, stay focused, Mr. Chisholm.
I'm sorry, my friend. How you gonna tell me what I am? The Lord forgive. Stop throwing stuff, Watson. Why you messing it up? You gotta go work with you something.
I would. I do many things by myself. You playing?
So you was messing all my work up.
No, why you did that? You don't put that on.
What are you talking about? No, just don't. No. Oh my God, Miss Brand, come here, cause she about to make me give her one of her face right here. One here.
Then you not telling me what we did.
Look, why she gonna move?
Okay.
What I put right there?
Cause you didn't tell. I don't think it's supposed to be right there.
Y'all should have y'all own TV show.
My question to y'all,
Man and apple.
Miss Branch, I just got
You
that
know what
now.
have a heart attack?
what part of the procedure are you on? Which
I don't
number?
even know.
See, I took the paper from me.
Can
Look,
you do a
tell me-
lab
look at
without reading?
that. I was like, give me the paper. Tell me, give
So
You
when
never
you don't
asked me that.
understand and don't know where you are, you got to go. You got to ask the question and you got to refer back to the procedure. But you're coming in and you're just trying to pick up and go on. Is that gonna work?
No, ma'am.
No, that's
Get
not
lost
gonna
in the
work.
wrong thing.
But what?
That's like going off in the woods.
So
with
my
a man.
question to both of y'all, to this team,
Okay, team.
what part of the procedure are you on and what do you need to
I'm
do?
getting sidetracked. She needs to hush.
You don't know.
I'm still listening.
So how would you build a complementary strand? How would you build that?
Okay, you said what do this one had the original over again, so this longer, and we'll do this first one over again. But what did this right here do what it had to do with this?
Because he had to build a primer because it attacks certain parts of the DNA when it start making copies. And so that was just an example showing you which part that it was going to bind to attack. That's what.
You know why
So-
I couldn't tell you that, though, honestly?
You know the words?
No, because when Miss Branch explained that to me, I was like, huh? Because she was talking to me like science terms. She had
So
to break
you,
it down to
because
me
I
like,
use,
what match with this?
you couldn't put it in your own words based on what I said and what that said,
Yeah, I really couldn't.
but you understood what to do.
Miss Brand, he's coming.
They not going to water it down for you when you get the test. Huh?
Are we supposed to have 2 of these?
We copy, we do.
Yeah. Well, no, doesn't matter.
Okay, because I only see one.
Yeah.
Okay, so we do the flip, so we do that too?
No.
Okay.
Okay. I say let me check my time. Time for you to finish that complimentary strain. Okay, we're going to have to wrap this up. Again, it's a 2-day activity. So what I want you to do at this time, make sure that you put all the pieces back in the bag. I just needed to know, at this point, what did you get from this lab? I want something from every group.
The different, like, how you copy it.
It's like when you had to copy one of the primers and put it in the other strand that we were working with.
How easily a disease form if
It's not easy.
disease
Let him, go ahead.
form if you mess up the strand.
Thank you very much.
You took my idea. 'Cause
You should be able to come up with something.
I was gonna say,
You gotta write that down.
What were you going to say? You get
that
points.
You like missing, not actually missing, but messing up, like something going wrong with the letters or having one wrong mistake. It could do some real bad. I wouldn't say technical because we're not electronics, but some bad stuff.
Yes. Okay, clean up at this time. Make sure all of the pieces are back in the bag. Sine, I need to hear something from y'all. Huh? All of the pieces, don't drop them on the floor. All of the pieces, look at you. All of the pieces need to go back in the bag. Huh? No, we're not going to finish this exclusive today.
So what is it?
All of the pieces need to go in the bag.
I need to go with the label that you just said.
Huh?
The label that is what I just said.
That's part of the data and the observations.
I can't say that because
I didn't know. I didn't know.
you take our lab books or no?
Put the lab books in the drawer.